we may start today's procedure, which is the defense of our student Viktor Mamontov. He will defend his PhD thesis, Escape Mechanisms of Mobile Genetic Elements Against CRISPR-Cas System and Diversity in Microbial Communities. We have a very distinguished jury, and in the order that we have on the slides, Professor Vestra is a professor in Environment and Sustainability Institute and in the Biosciences Department in the College of Life and Environmental Sciences at University of Exeter in United Kingdom. He got his PhD in microbiology in the University of Wageningen, and then was recipient of Marie Curie postdoctoral fellowship at the University of Exeter. He works on coevolution of phages and bacterial defense systems with specific focus on CRISPR-Cas immune systems, and also various systems of viral communication, synergy of antibiotics and phages, and interaction between the microbiome in general and antiphage defense evolution. Dr. Vestra, well, Professor Vestra is the recipient of several awards in the chronological order. It's the Westerbrink Award in 2013, the Westerdyk Award in 2014, the Heineken Young Scientist Award in 2016, 2 prizes from the Microbiology Society in 2002, and he also was a finalist of the Blavatnik Awards for Young Scientists in the United Kingdom. Professor Mojica, I don't even think I have to introduce him because he's the man who has coined the term CRISPR. Nevertheless, Professor Mojica is at University of Alicante in Spain, where he got his PhD 30 years ago, then continued his research in the United States at the University of Utah and the University of Oxford in the United Kingdom. And for like quarter of a century, Professor Mojica works at the University of Alicante. He was actually the first to observe that short DNA repeats in prokaryotic genomes provide immunity against phages and plasmids. And I mean, overall, the discovery of bacterial immunity was, adaptive immunity was a process, started with some statistical observations, some experiments, evolutionary studies, and eventually technology applications. And Professor Mojica was one of key players in this process. The awards are not listed here, but I assume that there should be a lot. Dr. Jackson is a senior research fellow at the Department of Microbiology and Immunology, University of Otago in New Zealand. I take it that it was quite difficult to compile a jury today because of time differences. He got his PhD there, and his research interests also focus on interactions between bacterial viruses and their hosts, and specifically about CRISPR-Cas systems, adaptation, discovery of new bacterial defense systems, and specifically relationships between phage defense and symbiosis in rhizobia. He also was awarded a number of prizes. He holds Sir Charles Hercules Fellowship for study of phage therapies, and he was awarded the Illumina Emerging Research Award as the top New Zealand molecular biologist. And now we have jury members from Skoltech, Professor Shidakov to the left of me, He's a full professor here at Caltech, and also he has a number of labs elsewhere, including Institute of Bioorganic Chemistry of the Russian Academy of Sciences, Pirogov Medical University, and a group at Central European University of Technology in the Czech Republic. He has many activities, but his main research is on analysis of repertoires of T-cell receptors and antibodies, the role of clonal populations in autoimmunity and cancer. He has recently published, well, among many others, but he was the lead author of a very important paper about identification of a clone, of specific clone, and development of drugs against this specific clone, leading to curing bacterial disease in a patient. And it's just a great paper and probably a seminal paper for a number of, well, for therapists to develop. 7 years ago, he was awarded the title of Professor of the Russian Academy of Sciences. And I think he's too lazy to apply for a full membership there, or too busy probably. Myself, I'm a full professor and director of Center for Molecular Cellular Biology here at Caltech. I work in various areas of molecular evolution and systems biology. In bacterial area, my main field of research is analysis of coevolution of transcription factors and their recognition sites, analysis of evolution of regulatory networks in bacteria, and also analysis of microevolution of bacteria on the level of strains, like genome rearrangements, phase variation, and things like that. The information here on the slide is somewhat obsolete. Indeed, I'm an elected member of Academia Europaea. I'm not an editor of Journal of Bacteriology anymore. I'm biology director as well. That doesn't matter. I'm a proud member, I'm proud fellow of International Society for Molecular Biology. Viktor's supervisor, Professor Severinov, is professor of molecular biology and biochemistry at Rutgers in New Brunswick in the United States, and head of laboratory of the Waxman Institute for Microbiology. His main research area is CRISPR adaptive immunity in bacteria, interactions between phages and bacteria, various molecular mechanisms that are involved in these interactions, analysis of natural peptide antibiotics. And recently he got interested in human genetics, and he heads the Russian initiative to sequence 100,000 genomes. And our candidate, Viktor Mamontov, he's got his master's degree from Faculty of Biology of the Moscow State University in 2017. And since then, he was working with Professor Severinov. In 2018, he joined the Caltech PhD program in life sciences. His project is about interactions between mobile genetic elements and CRISPR-Cas, the CRISPR-Cas system of Escherichia coli. He also worked as a computational biologist at Laboratory of Metagenome Analysis here at Caltech, studying the microbiome diversity in the Arctic Ocean. And he has published 3 papers in peer-reviewed journals, participated in a number of international conferences, and obtained a patent for 2 probiotic E. coli strains. So all formal requirements of Caltech are satisfied. And Viktor, that said, the floor is yours.
Can you hear me? Can you hear me well? Okay, let's start. Um, Good afternoon, dear jurists, dear colleagues and friends who attend online. Today, I'm going to tell you about my PhD results that conducted under supervision of Professor Konstantin Severinov here in Caltech. And the topic of my thesis is escape mechanisms of mobile genetic elements against CRISPR-Cas system and diversity in microbial communities. Okay. And as the main results of my research project are related to CRISPR-Cas systems, so let me shortly introduce to CRISPR-Cas systems. CRISPR-Cas system is an adaptive immune system of prokaryotes that provides them a defense against invading mobile genetic elements such as viruses and plasmids. And CRISPR is an acronym from clustered regular interspace short palindromic repeats, and Cas, it is CRISPR-associated proteins. And the first stage of CRISPR immune response is an acquisition of short DNA fragments into CRISPR array. And in the results, a CRISPR array extended by one spacer and one repeat. And then this CRISPR array is transcribed with the formation of precursor CRISPR RNA that is processed into short CRISPR RNA. And this CRISPR RNA bound with the Cas protein forms the CRISPR effector complex. And the main classification of CRISPR-Cas system is based on the structure of this CRISPR effector complex. For example, for class 1, it is a multi-subunit protein, and for class 2, the CRISPR effector complex is one single protein. This CRISPR effector complex, guided by CRISPR RNA, provides target recognition of invading mobile genetic elements or a protospacer that match to CRISPR spacer. And after this perfect match, this CRISPR effector complex conduct the degradation of these foreign mobile genetic elements. Excluding the active mechanisms to escape CRISPR interference, like anti-CRISPR protein, and generally mobile genetic elements can escape CRISPR interference by an acquisition of escape mutants in the protospacer region. But in this case, for example, like it's shown on panel B for type 1E CRISPR-Cas system, is it possible for CRISPR-Cas system to acquire new spacers during a primed adaptation? And during our research, we found out that's not only escape mutations are responsible for escaping of CRISPR interference, and there are some additional mechanisms that provides for mobile genetic elements to escape CRISPR-Cas system. And my study was focused on the investigation of the interaction between mobile genetic elements and type 1E CRISPR-Cas system. And on this slide, you can see the research objectives of my thesis, and chapter 2 is related, as I mentioned before, about the interaction, the mobile genetic elements and type 1E CRISPR-Cas system, and chapter 3, where I conducted computational analysis of assessing commercial kits for DNA extraction. And for chapter 4, I was responsible for genome assemblies and its annotation for subsequent in vitro and in vivo analysis. And let me start from chapter 2 about persistence of plasmids targeted by CRISPR interference in bacterial populations. And the story started from the transformation of model E. coli strain that bears an engineered type 1E CRISPR-Cas system with 2 inducible promoters. And for the transformation, we used plasmids bearing protospacer perfectly matched to CRISPR spacer. And induced cells we called CRISPR on, and non-induced cells we called CRISPR off. And after transformation, CRISPR on transformed cells were plated on media supplemented with both antibiotic and inducers to maintain CRISPR interference conditions. And CRISPR off cells were used as the control of the transformation, and they were plated on media supplemented with antibiotic only to select successful transformants. And on panel B, you can see the results of this transformation. And for CRISPR-on cells, we observed a drop in the efficiency of transformation, at least in 2 orders of magnitudes compared to the CRISPR-off cells. And the results are similar to different types of plasmids. For the plasmid PG8 with ampicillin resistance marker, and for plasmid PRSF with condomycin resistance marker. And next, we focused on the investigation of these CRISPR-on colonies and to rule out possibilities of acquisition, any escape mutations in plasmids. We extracted these plasmids from the original CRISPR-on transformants, and we used them for second round of transformation of freshly prepared competent E. coli cells. And actually the results of the new transformation were similar to the initial transformation. That means there were no escape mutations in these plasmids. And to test CRISPR activity in the original CRISPR-on colonies, we used these CRISPR-on colonies to prepare new competent cells from them. And for the transformation, we used compatible plasmids to check the activity of CRISPR interference in these newly prepared competent cells. And actually the results of transformation are pretty similar to the previous one. That also mean the CRISPR-Cas system remained active in the original CRISPR-on colonies. And for next, we were interesting about how long it's possible for CRISPR-on colonies to survive under CRISPR interference conditions. And for that, they conducted several rounds of resets into selective media. Orange media was implemented with both antibiotic and inducers to maintain CRISPR interference conditions, and yellow one was supplemented with antibiotic only to select plasmid bearing cells. And the gray, it is AB agar to calculate colony-forming units in original CRISPR-on colonies. And after the transformation and the first reset, we also conducted a real-time PCR analysis to estimate the number of plasmids per cell. And on panel B, you can see the results of these reseeding experiments, and the numbers above the bars demonstrate the results of qPCR analysis. And for example, for CRISPR-off cells, you see that the average compound number of such plasmids, it's about 200 per cell. But for CRISPR-on, we see this number is less than 1. That means the majority of CRISPR-on cells are plasmidless. And the same results we obtained for another type of plasmids. And we were wondering how it's possible for these CRISPR-on colonies to survive under antibiotic conditions. And to investigate the structure of these colonies, we cloned fluorescent marker into our target plasmids and repeated the transformation experiments. And on the first round of transformation, we revealed that CRISPR-on colonies, after transformation, there are two types of such colonies. One of them were fluorescent and the second one were dim, but the fluorescent colony were about 5% of the total number of colonies. And we tested these colonies by sequencing, and the sequencing results actually demonstrated that there is no genetic mutations in G8 proteaspace and in CRISPR array, but there are genetic mutations in Cas genes, around Cas genes. That means this CRISPR-Cas system were broken in these fluorescent colonies, and the majority of cells in these colonies were plasmid bearing. And for subsequent investigation, we used only dim CRISPR-on colonies. And here you can see also the observation of CRISPR-on colonies. For example, on panel A, it's pictures from confocal microscopy of the age of the colonies. This orange line indicates the age of the colonies. And for example, for CRISPR-on, we can see a fluorescent scaffold surrounded by dark plasmid-less cells. And the similar situation we observed on the center of the colonies on panel B. And we hypothesized that this fluorescent scaffold is essential for colony survivability on antibiotic conditions. And if the next step was to estimate the ratio between fluorescent and non-fluorescent cells. And for that, we conducted flow cytometry analysis. On panel A, you can see the results of this analysis. For negative control without fluorescent plasmids, we see that the majority of all events locates on GFP minus gate or non-fluorescent gate. And for CRISPR-off, the majority locates on GFP plus or fluorescent gate. But for CRISPR-on, we observed the distribution of events skewed to the right. That means there are two different subpopulations in CRISPR-on cells, and the statistics demonstrated that it's about 10% of fluorescent cells in CRISPR-on population. And if we assume that one single cell is responsible for colony formation, that means for formation of CRISPR-on colony, it have to be fluorescent or plasmid bearing cells. And to check this hypothesis, we conducted a microscopy observation of cell division in microfluidic chambers. And here you can see the divisions of CRISPR-off cells, and the majority of CRISPR-off cells remain fluorescent over the whole period of the experiments. But the opposite situation we observed for CRISPR-on cells. And after several divisions, the cells started to lose their fluorescent because of CRISPR interference. But the small fraction of CRISPR-on cells remain fluorescent. And if we track all divisions, we can build such tree with branches that demonstrate the fluorescent or non-fluorescent cells. And we hypothesize that it is important for colony formation to have at least one fluorescent cells to transfer plasmid from parent to daughter cells. And to explain the possibility of plasmids to survive under CRISPR interference conditions without genetic alteration, my colleague Alexander Martinov developed a mathematical model. And let me shortly introduce to this model because I think it's important to understand the whole picture of the research. The replication rate of plasmids is based on an equation of logistic growth, and it's shown by blue curve, and CRISPR interference rate is based on Michaelis-Menten dynamic, and it's shown by orange curve. And on panel A, we see that CRISPR interference rate is overcomes CRISPR plasmid replication rate on all plasmid numbers. That means in this situation, all cells should lose their plasmids quite soon. And on panel B, we see opposite situation where plasmid replication rate dominates under CRISPR interference. And in this case, the majority of cells remain plasmid bearing. And the most interesting situation on panel C, where there is a range of plasmid number, where plasmid replication rate overcomes CRISPR interference. And another question arises in this case: how it's possible for plasmid to reach this plasmid stability window? And we suggested that due to stochasticity of plasmid replication event and plasmid cutting event, there are some cells where plasmid replication rate occurs more frequently than plasmid cutting event. And if plasmid reach a minimum plasmid copy number in that corresponding to plasmid stability window, this system deterministically goes to stable state. But actually, this stable state might be broken due to cell divisions. And to test this hypothesis, my colleague Alexander Martinov also conducted a mathematical simulation for a plasmid with optimal copy number equal to 100 that presented on panel A, and panel B, C demonstrate the probability distribution to meet a finite number of plasmids in E. coli cells before their division. And this simulation were based on master equation. And on panel B, we started from just one plasmid per cell, and after several generations, the probability distribution converge into the same form. And the same form we also observed if we started from 100 plasmids, and for both situations, the local minimum though around plasmid equilibrium number that indicated by vertical lines. That means the form of distribution does not depend on the initial conditions, rather than the nature of plasmids and CRISPR-Cas system. And let me present also another important experiment where I cultivated CRISPR on cells in antibiotic-free conditions. For this cultivation, the culture were transferred into fresh media every 12 hours, and every 24 hours, the culture were plated on selective media to estimate plasmid bearing fraction. And we see a dramatical drop of plasmid bearing fraction for CRISPR own cells. But nevertheless, after 72 hours, it was still possible to identify some cells, E. coli cells, bearing the plasmid with wild-type protospacer and active CRISPR-Cas system. And we also noticed that CRISPR-off cells also have the decline of plasmid-bearing fraction, and we measured the growth rate for plasmid-bearing and plasmid-less cells, and we figure out that plasmid-less cells grow faster compared to plasmid-bearing cells. That means the plasmid provide additional metabolic cost for E. coli cells, and in these conditions, it's not beneficial for E. coli cells to keep plasmids. Another experiments were conducted with phage M13 to compare the behavior of phage with plasmids. For these experiments, I used a replicative form of phage M13 carrying a GFP marker and ampicillin-resistant gene. And I repeated the transformation and several rounds of reseeding experiments like it was done for the plasmids. And if I made some fluorescent colonies, these colonies were analyzed. And here you can see some results from these experiments. And actually on panel E, we see the two types of transformation for wild type replicative form, and on the right side, the transformation for replicative form with escape mutations. And similar to the plasmids, we see a drop in the efficiency of transformation for wild type of phage, and there is no significant difference in the efficiency of transformation for mutant form of phage. And on panel B, the results of reseeding experiments, and we observed the same situation for wild type and mutated form of phage. And several random E. coli colonies that were fluorescent were selected to test CRISPR adaptation. And we found that the majority of these samples, except of sample 6, contained the extended CRISPR array that suggested this CRISPR-Cas system provided immune response against mutated form of phage M13. And M13 genomes were extracted from these samples and sequenced. And results of sequencing revealed that there were different mutated genotypes in one colony that suggested that these mutations might be aroused independently during infection cycles. And let me present the conclusions of this research with the comparison mechanisms of escaping for phages and plasmids. On panel E, we see the infection cycles for M13 phage, and we can assume for the initial time of the first time, it is possible for phage to survive and thanks to replication dynamics, as it was shown for the plasmids. But if escape mutations simultaneously arose, this mutated form phage can quickly proliferate among cell population. And for that, CRISPR-Cas system have to respond by primed adaptation. In contrast to the phage, plasmids cannot proliferate horizontally, only vertically from parent cell to daughter cells. And if escape mutations arise in the plasmids, in this case, we didn't observe some significant impact on the E. coli population. And shortly, the main conclusions, we demonstrated the persistence of the plasmids in E. coli population without any genetic alterations and CRISPR interference conditions. And we also showed the heterogeneity in CRISPR-1 colonies. And we demonstrated the independent appearance of M13 mutated form during infection cycles. And let me also present additional results what I was conducted as a computational biologist. And in these studies, I'm not a first author, so let me present only introduction section and the results, what I conducted or where I participated. And for this research, my colleagues extracted samples from different environmental conditions, from water, ocean sediments, and digestive tract of Megalana gigas. And we tested 8 commercial kits for DNA extraction to identify what kits are better. And we tested different features, but I was responsible for computational analysis of 16S ribosomal RNA sequence data. And for this analysis, I used this pipeline, what shown on the right side. And first of all, I estimated the contamination level of in samples and in the control. For sediment and gut flora, we used non-input control, and for water, we used molecule control. And actually, we can see that all kits are different in community composition, even in control. And to estimate contamination level in samples, I used the Contam tool with very conservative parameters. I use method prevalence with threshold 0.5. That means if this operator taxonomic units is prevalent in control sample, it should be considered as a contaminant. And there you can see the visualization of relative abundance after filtering of contaminants. And you can see that here some free technical repeats per kits. And we see that kits are really different to each other. And it's also, for example, you can see on gut flora and monarch kit, it demonstrated like that technical repeats are significant difference to each other. And we also can see it on beta diversity plot on right side, that samples are quite far to each other. And for next analysis, we tested the reproducibility. On panel A, you can see the reproducibility per kit for technical samples, technical repeats, and it's also demonstrated some low reproducibility that some challenges for investigation. And on panel B, we can see the distribution of different types of OTUs between all kits. And in ideal situation, we would expect that kits should be shared unique universal OTUs among all kits. And it's also demonstrated on panel C. So we see that only short ratio of OTUs shared between all kits. And on panel D, I also conducted alpha diversity analysis for different kits, and in conclusions, the best kit should demonstrate high alpha diversity and low contamination level that presented on scatter plot on panel E in upper left side. And to conclude, we tested all features together for all kits and ranked them using min-max normalization for all features. And actually, PowerSol, PowerFecal, and LS Bio demonstrated the best scores for DNA extraction between all samples. And let me present the results where I conducted in silico analysis for bacterium producing E. coli strains with potential probiotic properties. From my colleagues, I received 10 samples of E. coli strains for assemblies, and I conducted these assemblies from long reads using Flyer tool and provided also error correction by Medaka. And I conducted the annotation of assemblies, and searching for biosynthetic gene clusters was conducted by anti-smash. And on right table, you can see the statistics of assemblies for the main strains that were selected for subsequent in vitro and in vivo analysis. And it is not noticeable that these strains also contained additional conjugates, and we revealed that these conjugates correspond to the plasmids. And here you can see the visualization of annotations of these strains, and it's worth to tell that these colicins that we revealed during bioinformatic search locate on the plasmids. So the plasmids provide additional benefits for these strains. And you can see the information about these strains in GenBank, what presented on these slides too. And the main results of my work in this study, the pipeline was for annotation of long read assemblies were developed, and the selection of appropriate candidate for subsequent in vitro and in vivo analysis were conducted. And the summary of all my thesis, we demonstrated the persistence of plasmids in E. coli cells with active CRISPR-Cas system. The commercial kits for DNA extraction were assessed, and novel bacterium producing E. coli strains were sequenced and annotated. And here you can see the list of publication where these results are present, and the list of conference where I presented the results from chapter 2. And I would like to make acknowledgements. First of all, I think my supervisor, Konstantin Severinov, for providing the opportunity to conduct these researches. And I also thank to my members from my scientific committees, Dr. Ispalatov and Dr. Lukyanov, for helping me during my PhD program, and my colleagues from Institute of Molecular Genetics, Dr. Evgeny Klimuk, Dr. Olga Musharova, and Dr. Ekaterina Savitskaya, who tragically passed on the start of my PhD, and Dr. Natalia Morozov for helping with microscopy, and Dr. Dmitry Sutermin, who helped me with computational analysis, and my colleague, Dr. Alex Martynov and Dr. Artem Isayev. Thank you for your attention and the question session.
Yeah, you're fine. Within time. Okay, so now we have the questioning session, and I would ask the jury members to ask their questions probably in the same order. So we'll start with Professor Westra, please.
Right, thank you very much. Thanks for the presentation. It's a very interesting thesis. I enjoyed, reading the thesis as a whole, but of course my own expertise sits most closely to the work that also forms the majority of your thesis, which is chapter 2 about the CRISPR plasmid interaction. And I guess, I just want to start with a really general question. Given your findings, how important do you consider CRISPR systems to be in shaping horizontal gene transfer? Can you just give us your views on that?
Thank you for your question, Dr. Westra. I think it's a really important question because there are many papers about it, and sometimes they have contradictory conclusions. But in my point of view, in evolution, from evolution point of view, I think CRISPR-Cas system does not restrict horizontal gene transfer. And I also think this to get some beneficial genes, it's depends on the environmental conditions. Because in one environmental conditions, these genes might be very helpful for prokaryotes, but in other, it might be provide just additional metabolic cost, and it in the best case, it will be eliminated from cells.
Mm-hmm. Okay, great. So that brings me to a question I had when I was reading your results. It made me wonder, how sensitive is the responses that you observe to the environment? And I was specifically also thinking about the concentrations and the types of antibiotics that you used in your study. So how does that influence the outcomes, you think?
Well, in my opinion, when we used media with antibiotic, of course, it was beneficial for cells to keep these plasmids to avoid dying from antibiotic. And for example, when I used antibiotic-free conditions, in this case, the majority of cells were plasmidless, and also I revealed some escape mutations in the plasmids too during this experiment that we didn't observe during colony reseeding experiment. And and in these both experiments, the conditions were very different on plate and in liquid media. And it's, for example, it depends on the area of research. And I think for medical field, it's more important to see the some environment on plates, because there is some biofilm structures that prevent to move antibiotics molecule to inside the colony, and these plasmid-bearing cells might provide additional defense against antibiotic in plate conditions. But in liquid media, there is no this interaction between cells like we observed in colony.
So can we specifically zoom in on the question how the type of antibiotics and the concentration of the antibiotics would affect, the results that you observe? So in other questions, how general is, the result that you obtain, and how does that depend on the type of antibiotics? Thinking about, in this case, antibiotic that is degraded extracellularly, and you have these altruistic social effects where you have survivors, because there are resistant bacteria in the colony. That may change depending on the antibiotic, but also some antibiotics may have an impact on the activity of the CRISPR immune system, something we studied recently. So, I'm just wanting to hear your views on how general the observations are. In this case, you work with ampicillin at one specific concentration, but how does the outcome depend on the type of antibiotics as well as the concentration of antibiotics?
In our experiments, we used actually 3 antibiotics: ampicillin, carbapenem, and kanamycin. And first two actually had the same action, and kanamycin is different. It blocks the translation process in bacterial cells. And we also used some standard recommended concentration of antibiotics, but for this it's pretty high for plates. So that means only cells with plasmids should survive under these conditions. Actually, during receiving experiments, we don't see a significant difference between carbapenem, for example, and kanamycin. But so that means there is no significant dependence on antibiotic type for example, for CRISPR colonies survivability.
Okay, so in some sense, you would expect some differences, I think. For example, antibiotics that impact translation may also lead to a reduced activity of bacterial immune systems because their production is, for example, blocked. We also studied the effect of bacteriostatic antibiotics, and we found that, when you slow down sort of the host metabolism, then that will have implications for the speed at which mobile genetic elements can replicate, and that can have implications for how well immune systems can, deal with invaders. So I think there is perhaps, there are some interesting questions there that are worth exploring, and perhaps something you can allude to in your, in your discussion. But maybe that's for another day. I did wonder, so you were talking about the differences in the brightness of the GFP and your CRISPR positive versus CRISPR negative cells. I'm assuming this will have implications for the levels of antibiotics resistance that you would observe. Have you tested that? Do you see differences in MIC between CRISPR positive cells carrying the plasmid versus negative cells carrying the plasmid?
Well, you mean the efficiency, the level of fluorescence from CRISPR positive and negative cells?
Well, yes. I mean, you used the GFP assay there, but of course you've done the same set of experiments, looking at antibiotic resistance. And so I was wondering, have you quantified the levels of resistance that cells have that carry, the plasmid when they also carry the CRISPR system versus when they don't? Are there differences there? Because there are clearly differences in the copy numbers of the plasmids, and that may translate into different levels of resistance.
I see your question. I think we just did a measurement in microfluidic chambers, and I think it's presented in supplementary figures. of course, the fluorescence signal, what was demonstrated from CRISPR off cells, the higher than for CRISPR on cells. But we don't know how many plasmids is required for providing the protection against the antibiotic. We also figure out that these, for example, fluorescent cells are grouped together in colonies. And I think to understand the mechanisms and more detail how it is possible for example, for plasmid-bearing cells to block and the antibiotic movement inside the colony and protect, for example, plasmidless cells. I think there is need additional experiments to show this.
There's some nice work from the lab of Alvaro Stan Milan showing that bacteria can, when they have plasmid-based antibiotic resistance genes, they can evolve higher levels of resistance very rapidly through elevated copy numbers of the plasmid. So I think this interaction, in terms of the copy number effects of CRISPR, is very interesting to think about, levels of resistance that are associated with that. In your microfluidics experiments, I'm sure this is in your thesis, I just don't remember it right now, but was there antibiotics in the experiments as well, or did you not include antibiotics there?
What kind of experiments do you mean?
In your microfluidics single cell analysis.
I see. Yes, in these experiments we didn't use antibiotics in media because it was really hard to catch a cell in microfluidic chambers. Mm-hmm. We have only several successful chambers, and because some chambers the cells lost the fluorescent, and fluorescent cells eliminated from chambers very soon. So it was not so long observation. And about your previous question, I would like to add, I also did a transcriptomic analysis of CRISPR colonies in unpublished results, and I figure out that there is a significant changes in gene expression. And if we, for example, suggest that the elimination plasmids from CRISPR cells occurs gradually, it is provide additional time for cell to change the gene expression. A lot of genes were related to some multipomes that provide to some defense against antibiotics.
I see. So, regarding this issue of copy number, do you think that, and again, thinking about the generality of your results, so you used a plasmid here that has a very high copy number, over 200. So what do you think happens when you have plasmids with relatively low copy numbers?
Yeah, I think it's also important questions. We used high copy number and inducible CRISPR-Cas system to provide the expression a lot of Cas genes, and it really depends on how many CRISPR effectors we have in particular cell. And for example, in these experiments, I think I did several experiments with low copy number, and actually there is very low number of colonies, and we can assume what in this case there is no chance for low copy number plasmids to survive under these inducible CRISPR interference conditions. But in nature, the concentration of CRISPR effector complex is much lower than in laboratory conditions. So, and the copy number of plasmids is very different. So it might provide the replication rate might provide significant mechanisms to escape some defense systems in prokaryotic cells.
I see. So that's interesting what you're saying there, because I wasn't aware that of data showing that CRISPR expression is lower in natural conditions. Can you elaborate on that? You know, the studies that show this.
Well, I know one study from Brown's laboratory, but I'm sure they used a natural CRISPR-Cas system or inducible, but they demonstrated that, for example, they also used some plasmids, and they demonstrated for against to these particular plasmids 50 CRISPR effectors provide about 80% of defense against these plasmids.
Right. Okay. I think the issue of you know, CRISPR expression in natural environments is probably one that hasn't been addressed very thoroughly. And it may well depend also on the system that one studies, right? There will be a lot of variation there. So I think it's very difficult to make these kinds of you know, to make these kinds of general conclusions. What we do see with our system, for example, is that expression tends to go up as nutrient availability goes down. This is not published, but it's something that we observed. And I think there are a lot of environments in nature that are much more nutrient limited compared to, you know, a flask with LB. So, you know, in our case, for example, I would expect that expression may often be higher in natural environments compared to, anyways, that's sort of, you know, a fairly minor point. But I did wonder, based on what we just discussed, you know, that interaction between CRISPR expression and plasmid copy number, do you think there is a coevolutionary interaction there potentially between CRISPR expression and plasmid copy numbers?
Yeah, I think it should be, from my point of view, because every expression, it's additional metabolic cost for cell, and in my point of view, it should be some normalized on the environmental requirements for cell, and some balance between defense and metabolic cost to between Cas expression. And as we see in, for example, from result from the chapter 4, where we annotated these E. coli strains, we also estimated the number of plasmid copy number per cell, and there is some dependencies between the size of plasmids and its copy number. So the bigger plasmids have lower copy number and opposite. And actually, we got about 90 plasmid copy number for the smallest plasmids were estimated by normalization of the number of reads per chromosome.
I see. Okay, so the final topic I want to discuss with you is about the you know, the link between your within cell modeling of the bifurcation of plasmid, you know, maintenance versus extinction versus population level dynamics. So maybe first on that subject, a very open question, like how do you actually do that? How do you move from that within cell model to population level model?
Yeah, I think it's a very important question, because it is easy to understand for one cell what's going on inside one cell. I mean, in relation to our model system. But for example, after division, we don't know where this copy number after division lies on plasmid stability window or not. And there is another round of replication between replication dynamics and CRISPR interference. But as we can see on this population level, it provides some significant impact. So we can see these cells with plasmids, but actually we also don't know about CRISPR interference rate. It's also might be different in some different cells. So, but for population level, it's it's significant impact between replication of plasmids and CRISPR interference rate. And
so in your experiments, what's the ultimate fate of the plasmid, you think, if you left this experiment forever?
I see. I think it's really depends on the environmental conditions. So what should we consider as transient conditions or permanent, final point of this dynamics? And from my point of view, I think the plasmids have to acquire and escape mutations to escape CRISPR interference. And because, for example, under antibiotic conditions, it seems all cells go to this final point where plasmids should get an escape mutations sooner or later, from my point of view. And but for initial stage of this period, the survivability of plasmids is provided by replication dynamics. But I think all plasmids without escape mutations sooner or later will be eliminated by a CRISPR-Cas system.
Yeah, I think so too. So we cannot have the stable coexistence of a CRISPR positive cell and a target plasmid, even in the presence of antibiotics, because as you point out, they will be eliminated. So either the plasmid needs to acquire a mutation, or the CRISPR system needs to be mutated. And probably, so this kind of links back. So this is the with antibiotic scenario, and I was also thinking that if we imagine a situation where we have cells with or without CRISPR carrying the plasmid, probably the cells without CRISPR have higher antibiotic resistance levels because they have a higher copy number of the plasmid. So at high concentrations of the antibiotic, I assume that the CRISPR positive cells will be quite quickly outcompeted. This is something you didn't measure, but it's I think that would be my prediction. So we don't have the stable coexistence there because these cells are simply replaced by cells that don't have CRISPR, or as you suggested, mutated plasmids that can coexist with a CRISPR positive cell. In the absence of antibiotics, I also find it difficult to see how you could have this stable coexistence. And actually, if we look at your, figure 2.5, in the absence of antibiotics, you can see that the frequencies of cells carrying the plasmid are going down quite quickly over a couple of hours. It goes from 100% plasmid carrying cells to maybe 25%, and then to maybe 10%. So you can see that there is a steady decline in plasmid carriage. And so my interpretation of that is that it's a very interesting transient dynamics that you observe here that could have actually important long-term implications, because it allows for these mutation events to occur that you just mentioned. So I'm not saying it being transient means it's unimportant, not at all, but it's probably not something that would be stable over long periods of time, even though your within cell modeling suggests that this is a stable state. I think at the population level, that's not the case. Do you agree with that sort of assessment?
Yeah, I think I agree with it. But I also think if they, for example, if they exclude a possibility of mutations, and if the cell, for example, cannot divide, and we look at only one single cell, and if plasmids locates in this plasmid stability window, and so from my point of view, it's not possible for CRISPR-Cas system to eliminate the plasmids because of the rate of CRISPR interference just lower, and mathematics showed us this stable state. But due to there is a probability to get mutations, and cells permanently divide, these stable conditions is permanently broken, and there is some interplay between dynamics rates.
Thanks very much. I'm going to hand over to one of the other referees. Thanks for this discussion.
Thank you, Professor Vestra. Before we move on, I have to ask a formal question whether you are satisfied with the responses of the candidate.
Yes, yes, yes. I am completely satisfied with the responses. Thank you.
Okay, thank you. Okay, Professor Mojico, please.
Okay, thank you. First of all, thanks for the nice presentation, and also congratulations for the interesting work you have done. One of my main questions was related to one that Professor Vestra already did, is these aspects of what happened in nature compared to the experiments in the laboratory regarding the copy number, the high copy number of the plasmids. So I'm satisfied with your answer to that point. And anyway, I really enjoy reading the thesis, but I must admit that I had, let's say, a hard time trying to understand the rationale for this missing inactivation of the CRISPR-Cas system or accumulation of the escape mutations in the protospacer as the cause of the apparent plasmid maintenance in CRISPR-on colonies, especially since you are working most of the time, okay, with mixtures of cells from colonies that might contain these kind of mutants, albeit in a very small proportion. Again, eventually, after reading this chapter a few times, this chapter 2, and thanks to the clarification you have introduced in the text, I think I already got it. Okay, not completely sure, but I think that I now have it. So, however, let's ask me one question related to this. In the section 2.33 on the direct observation of plasmid bearing cells in CRISPR-on colonies, you conclude that for continued colony growth on selected medium, there must be at least one uninterrupted line of plasmid bearing cells that persist through multiple generations. So my question is, could these uninterrupted lines of fluorescent cells only be descendants of mutants with either an inactive CRISPR-Cas system or with escape mutations in the target?
Well, thank you for your question, Professor Mojica. Well, I think it is possible because we work with mixture of cells, and we observed only, I would say, indirect proofs that there is no mutations in the plasmids, but I think it's also possible, for example, for colony formation, if in all lines of plasmid-bearing cells, for example, will be eliminated by CRISPR interference, and only one line will remain, but in this line, the plasmid were mutated, And in this case, it's also responsible for colony formation. But I think in this case, if we picked up this colony and reseeded it on selective media, we should observe the majority of cells should be plasmid bearing under conditions with both antibiotics and inducers. And we also might be observe a fluorescent from these colonies, like for CRISPR-off cells without CRISPR interference. So yeah, I met some these colonies, but it was very rare events. And as we conducted the sequencing of similar colonies from transformation, and we find out the mutations in CRISPR-Cas system. So I think it's possible. And if I met these rare colonies on my experiments, I just ignored them because the majority of colonies was dim or not fluorescent. And for example, for next round of reseeds, I used only dim colonies. But also when I conducted this long-term cultivation antibiotic-free conditions, where I analyzed all mixture of in medium tube, for example, all variants, I also met mutations in the plasmids. So I think it's possible.
Yeah, I really think that it's not just a single experiment you did, but all the experiments all together, the main result altogether, all of them, that really shows that you have these cells with an active CRISPR-Cas system and with a mutation that are still there. Because if you look one by one, in itself, I get doubts in some cases, but altogether make a lot of sense. It's not easy. It wasn't easy for me, but probably it's my fault. So, okay, also regarding the chapter 2 on your published results on the transcriptome assay of CRISPR on cells, you didn't include that in your presentation. This is the section 2.3.6. You analyzed the transcriptome of E. coli transformants, you say, after cultivation in kanamycin supplemented medium. That's right. Which transformants are you referring to?
Yes, actually, these colonies were cultivated in liquid LB medium supplemented with kanamycin, and I think the problem of this analysis was that there is no appropriate control from my side. I used the only CRISPR-on and CRISPR-off cells, both these plasmids and antibiotics, but I didn't use for this analysis cells, for example, without antibiotic, but only with inducers. So that means we can't resolve the impact of inducers on gene expression analysis. But if we compare just CRISPR-off and CRISPR-on cells, we see that some genes, the changes in gene expressions are significant, and some genes are related to multi-drug resistance function, something like that, that provide, what might provide some defense or metabolic exchange in bacterial cells to help them to survive under antibiotic conditions. So, but for this study, I think we need some additional control for more appropriate conclusions.
Okay, so the transformants themselves were carrying which plasmids all the M13?
Yeah, these plasmids.
The two plasmids?
No, only for one.
Which one?
With economicin.
Economicin. Okay, it's because in the text it's not written. So it's because it's one section where you talk about the M13 that it's not clear which kind of transformants are you talking about. And
yeah, so
also related to that, after in that section also regarding the M13, you select the cells that have acquired this plasmid, the replicative form, I mean. You select them with antibiotic.
yes, this M13 replicative form also carries penicillin resistance gene.
Penicillin, I thought it was economicin. Yeah, I saw in your presentation that you talk about ampicillin, but at some point I thought it was economicin because in fact it's not included also in the text. You missed that.
I see.
Yeah. Okay, let's jump to chapter 4. In chapter 4, mini-ion was used to obtain complete genomes of 2 E. coli strains. And it seems that you are very happy with mini-ion. And I got very contradictory inputs from people that work with mini-ion. So just how reliable are the sequences you obtain? Because I guess you perform the genome assemblies. I assume you check the quality of the sequences. How happy really you are with mini-ion?
From my point of view, I like mini-ion because it's very convenient to produce a DNA library, and the sequencing procedure is very easy and fast, and it's improvement every year. The technology is improvement, and the results, for example, for single strains, are very nice results. All contexts are circular, so you don't need to some additional computational approaches to resolve some these nodes and graphs. So for me, it's very nice for sequencing some single strains. And yeah, but the accuracy is not so good like for Illumina or PacBio, but it might be resolved with higher coverage.
High coverage, yeah, absolutely. Yeah, but it's improving very fast. The chemistry of the mini-ion a few years ago was terrible. Okay, I'll say after the changes you have made, there are some inconsistencies through the text, so relating, for instance, to the use of type and subtype when referring to the CRISPR-Cas systems. They are written in both in upper and lower case, and sometimes you refer to subtypes as types. There is in the abbreviation list, you talk about the track RNA instead of the tracer RNA, and you define it as transacting instead of transactivating. But later in the text, this is correct all the time. So I think you could change that easily. It should be. And but in general, I'm very satisfied with how you addressed the comments and questions raised in my review, and also with the answers during this discussion. Thanks a lot. And congratulations again.
Thank you, Professor Mojica.
Okay, and then again, I have the same formal question. Professor Mojica, are you satisfied with the responses?
I do, very satisfied.
Okay, thank you. I'm sorry, it sounds silly, but it's formal procedure that we have to follow here. Okay, Professor Jackson.
Yeah, hi, Victor. Congratulations. Yeah, very nice work. Enjoyed reading it and your presentation today. So I thought I might dig a little bit more into the mechanics of what you think is occurring. So you've got this model where there's kind of a competition state between the replication versus the interference of the plasmid. And if we're assuming we're not progressing to the ultimate fate of acquiring a mutation, as you discussed with Edza, let's assume the plasmid doesn't ever mutate. I'm wondering what contribution you think priming would have on how the plasmid is actually lost from the cell in the absence of mutation.
Thank you for your question, Dr. Jackson. Well, as I know, primed adaptation is significant if these target mobile genetic elements obtain escape mutations. Well, I didn't focus on the primed adaptation, but from my point of view, yeah, we didn't. I think my colleague from Olga Musharova conducted this project to estimate primed adaptation against different PAMs, and actually, yeah, there were primed adaptation against mutated form of plasmids.
Are you familiar with the studies showing acquisition of spaces from Cas3 derived fragments in the absence of any mutations in the target?
Sorry, could you repeat your question?
So for example, there are several reports that you can acquire spaces via primed adaptation from interference proficient targets without any point mutations. Are you familiar with these studies?
Well, as I know, it's really depends on the type of mutations in the prosperous region, and for one type E CRISPR-Cas systems, there are two different some pumps that provide normal CRISPR interference, AAG and ATG, but they have actually different strange for CRISPR interference. And I think it also depends on the number of mutations in the protospace region. But well, but I know about these researches only from my lab about primed adaptation.
Do you know where the substrates that are acquired as protospaces generally come from during primed adaptation?
Well, mostly it's come from non-target strand during primed adaptation.
I'm just wondering if you can maybe link Cas3 to the question.
Sorry, what your question about Cas3?
Okay, there's some studies come from Konstantin's work and also from Stan Brunn's showing that fragments from Cas3 are preferentially acquired as substrates, as new spaces, and that this can occur in the absence of target mutations as well. So what I'm wondering is what role adaptation actually has in your model, and whether you would expect a similar outcome if you perform the same experiments in the absence of Cas1 and Cas2.
Yeah, I see. Actually, for dynamics modeling, we didn't use the possibility for primed adaptation. And in our system, in our type 1A CRISPR-Cas system, we have all machineries including Cas1 and Cas2. But for these experiments with plasmids, we didn't observe an CRISPR adaptation. Actually, we tested several randomly chosen colonies, 10 colonies after transformation, and we didn't observe the extended CRISPR arrays. But in contrast to the plasmids, we observed the extended CRISPR arrays in the experiments with M13 phages.
Okay, maybe just moving on. What do you think the effects of CRISPR regulation? So, for example, studies showing that quorum sensing can upregulate CRISPR in some hosts at high cell density, and how that might affect plasmid copy number and the potential benefit or detriment to the host under different scenarios of regulation.
Well, in our laboratory conditions, I think it would be possible to use different concentration of inducers to regulate the activity of CRISPR-Cas system. I mean, the expression of Cas genes. As I know, in nature conditions, it's not always Cas genes expressed, and sometimes it's expressed against the invading mobile genetic elements. And for example, in E. coli, one type E CRISPR-Cas system started, it is inhibited by NHS gene, and if we did the knockout of the gene, CRISPR-Cas system started to express. And yeah, and as I know, there are many others defense systems, and they can work in combination. But as I know, CRISPR-Cas system is essential on the first stage of infection by phages.
Yeah. Okay. Thanks. So based on what you've kind of learned in your study, you made a comment in the discussion about CRISPR-based tools to clear AMR plasmids, for example, in the context of treating a patient. What are the real take-home messages, or how does your study inform how these types of therapies should be developed?
As I know, there are several commercial companies and methods using CRISPR-Cas system, for example, to eliminate the plasmids with bearing antibiotic resistance genes. And I think we should take into account the information about copy number of plasmids, and it's better to choose a different proteaspacer target for these approaches to be more convincing more about successful results of this therapy.
So do you think your data shows that it's ultimately a futile effort if the plasmids can still persist, or do you think the therapies will actually work?
Your question about persistence of plasmids in
the context of these therapies which are using CRISPR targeting, your work shows the plasmids can still persist in the population.
Well, I think it depends on what tool we can use for the delivery of CRISPR-Cas system, into the cell population. Sometimes it might be challenging because, for example, big conjugation plasmids can also have additional mechanisms to defend themselves against CRISPR-Cas interference, for example, bearing anti-CRISPR proteins So it's also should be considered in this type of therapies.
I mean, I think in Ed's line of questioning, you decided that the ultimate fate of the steady state was to collapse to a point where you'd acquired a point mutation that would then evade targeting. So if we assume that would be the ultimate fate in such a therapy, what potential strategies might you use to overcome that being the final end state?
Well, from my point of view, the targeted mobile genetic element should be well annotated to understand what genes it bears that might provide additional challenges for CRISPR-Cas system, and the should choose that CRISPR-Cas system. For example, if these mobile genetic elements has anti-CRISPR proteins, that such CRISPR-Cas system that is not affected by this anti-CRISPR protein, for example.
I mean, for example, if you had two spaces targeting the plasmid, would you expect more cells to completely clear the plasmid versus acquire point mutations?
In my point of view, it's more, it's better to use several spacers for targeting, because if one space, protospace is mutated, at least one of them should work for CRISPR interference. And from the point of view of my research, I think if we, for example, if they used additional spacers, it doesn't provide it doesn't increase the CRISPR interference rate, because we have CRISPR interference complex. Its concentration is the same for one CRISPR space or for two or three. It doesn't matter. But for targeting, it's better to use several spacers.
Yeah, I think there would be some really fascinating modeling to do to look at escape and priming in your system with a view to actually trying to prevent the frequency of escape mutations versus clearance of the plasmid from the population. I think there's a lot of interesting stuff to explore there. Yeah, and similarly, most of your work is focused on type 1 CRISPR. I wonder if you might had any general thoughts on how it might apply, for example, to RNA targeting systems like the type 3?
Yes, I think it would be interesting to see the dynamics for other CRISPR-Cas systems, but I think it might be a little bit difficult because, for example, as I know, type 3 CRISPR-Cas system that targets RNA molecules, it's usually locates in thermophilus, so it's some additional difficulties for experiment design. But I think it's also should be depends on molecular dynamic between mobile genetic elements and CRISPR effector complexes on the first stage of observation.
I mean, I would argue that there are plenty of amenable hosts for studying type 3 systems, so that shouldn't be a factor. And it's sort of an interesting dynamic. Again, coming back to escape mutations, the type 3 seem to be a lot more tolerant of the mutations. So the end fate of the system, again, might be driven more towards complete elimination of the plasmid as opposed to escape plasmids arising in the population. So I think it's just worth thinking about kind of the longer term dynamics and not just assuming the steady state you've pinned on your graph is the final endpoint. But yeah, I mean, other than that, great work, and I think that's the questions from me.
Okay, and again, the standard question, whether you are satisfied with the responses.
Yeah, generally, yes, but I think there's some, and I alluded this in my comments, there's some key literature around priming that you probably need to familiarize yourself with a little bit more.
Okay, yeah, we can discuss that during jury deliberations.
Yeah, but otherwise, it's all great.
Okay, Professor Chudakov.
Yeah, thank you, Viktor, for the nice story and nice work. I should admit that, okay, my current specialization is in immunology, and I mean human immunology, not bacterial one. But in the previous years, I worked on fluorescent proteins development for like 10 years, and I'm always happy to see something fluorescent and different resolutions, which reminds me these beautiful stories. And especially this incubation in chambers, the microfluidic looked nice. And in these days, when we develop different fluorescent proteins, some of those bacteria didn't like, because at some expression level, there were probably some toxic effects. And they also these colonies got rid of the fluorescent protein expression. It looked quite similar sometimes to the pictures you are showing. It's like sector-like loss of fluorescent proteins expression, or some dim colonies which decrease expression. If you zoom in, it often looked like very similar to what you have shown, like some sporadic loss or preservation of expression. So it try to get rid of this expression. At the same time, the antibiotic pressure forces, and it somehow balances in between. And we never analyzed it, of course, because it wasn't of our interest, and because, okay, it's a complicated stuff. And it is still complicated stuff in your work. So for me, actually, the whole logic, how you were trying to analyze what is going on, what you're trying to rule out, in terms of escape mutations in the plasmid and damaging the bacterial mechanics by reseeding, by reproducing the plasmid, checking what is going on, etc. It's quite complicated for me. I thought that I was understanding it when I was reading the thesis. Now I'm a little bit lost again. Okay, if I dive back and all the details, I will probably understand again. But I want to focus on one particular aspect, technical thing in this story. So there is a colony growing, and it is clear that it is heterogeneous, right? So some cells are still fluorescent, some not. And it's quite, even though they could be lower fluorescent, but still it's quite like yes and no situation. So it is heterogeneous, and maybe it is even more heterogeneous inside. And then when you reseed it, this colony as a whole, or grow the plasmid from it or something, you tend to analyze what happened like in bulk, right? Like average temperature in the hospital, what we call. And at the same time, the colony inside is heterogeneous. I understand that it is technically hardly feasible to distinguish what happened at the level of small portions of the colony. And different colonies may have different escape mutations and different events, particular events behind what we observe. But have you tried, I see the flow cytometry in the thesis, but have you thought about like sorting maybe the fluorescent and non-fluorescent cells and reseed after sorting, get the plasmid, check, analyze, okay, ideally the single colony level, but I guess it's not feasible, but at least in bulk, but still try to separate the patients with different temperature. And could this give us some more hints what is really going on, considering this heterogeneity of the colony inside, which currently, please correct me if I'm wrong, but in the current way of analysis, looks like we are at some point forgetting about this, saying, okay, something happened in the colony, we'll reseed and analyze, but we forget about the heterogeneity at this moment. Or am I wrong? Then correct me, sorry.
Yes, thank you for your question, Professor Chudakov. Well, actually, we had this idea to select only fluorescent cells during fluorescent cytometry and reseed it on the selective plates. But the problem was, we used some specific buffer for bacterial cells. And when the did a selection, and it seems that it was a little bit harmful for cells after this procedure, and as we did in other institute, it was hard to then transfer these cells to our lab and provide manipulations with them. Well, I think it would be interesting to select only a fluorescent cell, and if it would be possible to, after selection, we need to reseed these cells on selective media immediately. So, and I think in this case, it would be.
Okay, let me rephrase it. Let's forget about sorting, some additional technical complications. Okay, but if you get one colony, just one colony, dissipated and reseed and culture, and then you still have some heterogeneity like this, like to have statistical information of what has happened inside a single colony. Did you try thinking this direction? And is it rational what I'm saying now, or maybe not?
But I think it's rational. Well, as we tried to do this, and I think we got really low amount of cells, and the concentration in the buffer was not enough for appropriate reseeding experiments, in my point of view.
I thought one colony is lots of bacteria, and you see them, and you have lots of colonies. No,
we had about I think 300,000 events from one single colony during these cytometry experiments. And if we assume that the majority of, we see only 10% of cells fluorescent, it's about 30,000 cells for reseeding experiment. So, but I think after this reseeding, these colonies should go to heterogeneous conditions. I mean, this fluorescent fraction after sorting.
no, I now just meant avoid sorting, just get one colony, reseed, and get then information about each of the colony grown. So focus on an analysis, genetic analysis of one single colony. What is heterogeneity inside? What could be the rules? Okay, that's rather as ideas for further discussion. Probably, I'm not quite sure the good ideas. So just discuss it later. Got it. Yeah. So some scope to think about how to measure it. So as I heard from you now, you also still have these hesitations, as I have. Yeah, that we are measuring afterwards, and the reseeding and analysis of the plasmid, average situation in these colonies, but not the heterogeneity of this situation. So, do you agree with this conceptual, like, let's call it drawback, that we don't know?
Yeah, I think after reseeding, these colonies during growth will be heterogeneous. So that means some lines of cells will lose their plasmids under CRISPR interference conditions, but due to dynamics, till some mutations arise either in the plasmids or CRISPR-Cas system, it provides some plasmid-bearing fraction in these reseeding colonies. Well, actually, I think it's very similar to that we did on during reseeding experiments. So because we assumed only fluorescent cells are able to produce viable colony on media supplemented with antibiotic and inducers, but it was, of course, it was indirect observation. It was like the results of our experiments. So But if they, for example, conducted the sorting of, for example, this colony before reseeding, certainly they collected only fluorescent cells, but I think after reseeding, they also produced this very similar colonies what we obtained during some normal reseeding experiments.
Mhm. Okay, thank you. I guess I'm satisfied with the answer at the level we could do it now. If I'm not going to continue the new levels of this research. So.
And are you satisfied? Yeah.
Yes.
You have to say that. Okay, and I should add that we've been in contact with Viktor because I was his PhD individual committee. So my comments have been taken into account at previous stages, and I'm happy with the thesis as is. So now, do we have any questions from the audience? I see no audience, so probably we have no questions.
Well, no, we do have, yes, one or two members of the audience, but I don't see any questions as well.
Okay, sorry. Oh yeah, there are more. Sorry, it was my mistake. There are more people indeed in the Zoom. Then Professor Severinov.
Hi, can you guys hear me?
Yep.
I forgot what the procedure is. Am I to say about the work, about Viktor, or what am I commenting on?
Basically, whatever you want, but it is my understanding that the main role of supervisor is to say about how Viktor worked, what was his individual contribution to the research, and
So,
not the scientific novelty and importance, but rather about his contribution.
okay. So I consider this a very good thesis, and Viktor has been a pleasure to be with for, well, I believe it's 6 years by now, even more maybe. From my perspective, of course, is the CRISPR study, which is the most significant for his thesis, because much of the later work was done because of the circumstances in Russia which were beyond everybody's control, which forced him to change the direction, let's put it this way. But the work that he published in PNAS as a first author, I think is an interesting, first of all, it's kind of an untrodden part of the CRISPR juggernaut, if I may say so, right? So this was an unusual study, and I think that's what makes it important. It's certainly interesting and potentially important given the possible effects, consequences to the understanding how CRISPR interacts with various mobile elements. And that particular work, which he literally did, I don't know, at least 90% of everything that was presented, involved many different techniques from microbiology to single cell monitoring to fluorescence sorting to ultimately mathematical modeling. And he clearly was the driving force behind all of that. He did master a lot of mathematical modeling during the analysis of his data that he did with Yaroslav Ispalatov, which allowed him to then smoothly move into bioinformatics and computational biology, which I believe he wants to study later on professionally and work on as a postdoc. But I think the basis in experimental biology and skills that he acquired during his work for the thesis would certainly be very useful for him in his future work. I think it's a great thesis. So I don't know if there's a grade for this, but he certainly deserves an A, in my opinion. That's it.
Thank you, Konstantin.
Okay. And now I think we have to go to deliberate in separate room. Yelena, would you please organize that?
Yes, so dear colleagues, we're
So
ready
the jury goes to
yes, so we're ready
deliberation room in Zoom, and Viktor probably will have to leave for a moment.
Yes. So I think at the moment, we have only mostly jury members left and Professor Severinov. So, dear colleagues, I will open a separate breakout room for the jury members now. Please join it.
Okay, Viktor, I congratulate you. You have passed with minor revisions, which means that you'll have to take into account some more comments. But there's no need to repeat the procedure. So I think that it's Dr. Mamontov from now on.
I would like to thank you all who attended my PhD defense, especially for my supervisor Konstantin Severinov and all juries jury members who agreed to take my invitation for this PhD defense, for their available comments and questions that makes me to look at my research from different sides. So thank you all.